Hello and welcome back to our session dealing with the cell death. In the last short video, we will have a closer look at the necrosis. We will compare necrosis to apoptosis, and we will also learn a bit about the recently discovered way of inducing cell death called necroptosis. As you can see here, necrosis is really different if we compare this cell death to apoptosis. Necrosis is an unregulated process, and necrosis is always pathological. It is primarily driven by external extrinsic factors, and examples of those include infection. Necrosis can also be initiated by direct toxic threat, by freezing, excitotoxicity, this is quite important in nerve cells, or trauma. So how does necrosis look like? As you can see here, necrosis is actually not very well regulated in the cell. And the first step is always a trauma. This can be an exposure to toxic chemicals, mechanical injury, heat, cold, or hypoxia. So in necrosis, cells lose the ability to regulate the internal environment. This leads to calcium influx, which is accompanied by swelling. And this is the first difference between necrosis and apoptosis at the cell level. In apoptosis, cells shrink. In contrast, in necrosis, they swell. And what you also should keep in mind that necrosis is always accompanied by influx of calcium. This uncontrolled calcium influx leads to alteration of protein activity, and here especially calpain and catepsin are important because they are getting activated by extremely high calcium levels. Once calcium enters the cell, this leads to release of additional calcium from endoplasmic reticulum, resulting in extremely high calcium levels. Subsequently, toxic compounds are formed within the cell, and this leads to cell death. Importantly, and this is also additional difference between apoptosis and necrosis, necrosis leads to uncontrolled release of cytoplasmic proteins into the surrounding, and therefore it always ends in inflammation. What you can see here is an experiment conducted in a dish. Here the cells have been fed with a calcium-sensitive dye. So as you can see here, the first step is always influx of calcium, and this cell, so here calcium enters the cell, and the cell is swelling, followed by uncontrolled release of the cytoplasmic compounds. So again, we have during necrosis the first step where calcium enters the cell. We have a swelling of the cell itself, but also swelling of the endoplasmic reticulum and mitochondria. This leads to release of additional calcium from the calcium stores in the endoplasmic reticulum. This leads to progressive injury, accumulation of toxic compounds within the cell, breakdown of plasma membrane, breakdown of organelles and nucleus, and finally leakage of the contents. As a result, there is inflammation in the surrounding of a necrotic cell. So, what are the hallmarks of necrosis in a nutshell? We have cell swelling, we have organelle swelling and damage, depletion of ATP, and I hope that you remember that the levels of ATP are kept in apoptosis. We have a loss of ion homeostasis, membrane rupture, nuclear lysis, cessation of protein synthesis. Necrosis is also irreversible and also promotes death of neighboring cell. So if we now compare both processes, then we will notice that there is cell shrinkage in apoptosis In contrast, in necrosis, there is cell swelling. Again, necrosis leads to depletion of ATP. In apoptosis, those levels are maintained. And apoptosis does not adversely affect neighboring cells, whereas necrosis promotes cell death of neighboring cells. In reality, there are even more differences, because what we have covered so far were merely morphological features. However, there are also very important differences in terms of physiological significance. Again, apoptosis only affects individual cells because it leads to production of apoptotic bodies. They are then removed by macrophages and also by neighboring cells. In contrast, in necrosis, we have adverse effects on group of neighboring cells. Apoptosis is induced by physiological stimuli, such as lack of growth factors or changes in the environment. In contrast, necrosis is evoked by non-physiological disturbances, including trauma, infection with the viruses, hypothermia, hypoxia, or metabolic poisons. In necrosis, there is phagocytosis by adjacent cells or macrophages. In necrosis, the components of the cell are taken up by macrophages, but not as vesicles. This leads to production of inflammatory signals by the macrophages, but we will look at this process closer in our immunology lectures. Importantly, necrosis always leads to significant inflammatory response. In apoptosis, there is absolutely no inflammation going on, and this process is very, very well controlled. If we look at the biochemical features, what we need to keep in mind is that necrosis leads to a loss of regulation of ion homeostasis. In contrast, apoptosis is a tightly regulated step and it maintains the homeostasis. Necrosis does not need energy. In contrast, apoptosis is ATP dependent. In terms of DNA fragmentation, in necrosis, this DNA fragmentation is mainly uncontrolled. In apoptosis, DNA fragmentation is well controlled. And lastly, the DNA fragmentation in necrosis is very randomly. After we have looked at apoptosis and necrosis, I would like to show you a newly discovered cell death mechanism, which is, however, not relevant for the exam. So this is, again, just to show you the bigger picture. Necroptosis is a cell death which combines elements of necrosis and apoptosis. So recent investigations have revealed that there is substantial crosstalk between apoptosis and certain forms of necrosis. And this newly discovered process have been termed necroptosis. Necroptosis is a form of programmed controlled necrosis that shares some of the features of apoptosis. It relies on molecular switches that act to make cellular decision between apoptosis and necrosis. And the regulation of this process is mainly maintained by receptor interacting protein, RIP kinases. With this, I would like to end our session on the cell death, and I will see you soon in our microbiology lecture dealing with parasites. Thank you very much for your attention.